Mosquitoes have been targeted by some of the world's most intense pesticide programs and, as a result, have come up with many strategies for avoiding control. In 1989, 114 different species of mosquitoes resisted at least one insecticide. Many have developed resistance to more than one insecticide, especially to the powerful nerve poison. Resistance comes from a number of different cell mechanisms that prevent the attachment of the insecticide to the insect's nerves. In some cases, an enzyme called esterase attaches to the insecticide before it gets to the nerve ending, blocking insecticidal action. When there is a lot of insecticide around, this strategy works like shoveling the snow in front of your house during a heavy snowstorm. It's successful only if you have a lot of shovels. But these mosquitoes have a lot of chemical shovels to remove the insecticidal snow because, not so long ago, they invented a way to increase the production of their chemical shovel many times over. Sometime, somewhere, while large quantities of nerve poison insecticides were first being used, the esterase gene was duplicated many times inside the cells of a mosquito. Many copies of a gene produce much more protein than a single gene. This overproduction increases the amount of protective esterase, which can make even massive quantities of the insecticide harmless. The new strategy was extremely successful, and from its first recorded appearance in 1986 quickly spread around the world. How did this mutation, i.e., the gene duplication, appear so suddenly and so widely? Two possibilities exist: that the mutation appeared several times independently or that it spread like lightning from one place. Careful research on DNA from resistant individuals of the mosquito Culex pipiens from around the world tells us that the DNA surrounding the duplicated genes is identical in mosquitoes from California, Pakistan, Texas, and Egypt. Because it would be almost impossible for the exact same mutation to happen in 4 different parts of the world, it seems that just one single mutation, a drastic one, caused esterase duplication in this species. Live adult mosquitoes fly internationally, hidden on airplanes, and easily leap across continents. So duplicated esterase genes soon went on a global tour, leaving offspring everywhere they stopped. But evolutionary changes such as those observed in the mosquito, like contracts with the devil, carry a cost. A great amount of raw materials is needed to make overproduced esterase, and insects that needlessly make so much of the extra protein are selected against. Field studies show that, in the absence of insecticides, mosquitoes without the overproduction of esterases grow faster, survive longer, and reproduce better than the resistant types, and that areas without heavy pesticide use have fewer mosquitoes with the duplicated esterase genes. Natural selection against resistant individuals reduces their frequency in following generations, as long as the insecticides are not used. Nevertheless, evolution can run subtly, driven by selection to reduce the costs of insecticide resistance, and mosquitoes that pay the devil a discount price can thrive. For example, most mosquitoes make too much esterase and wastefully spread it throughout their bodies into tissues where insecticides have no effect. Other mosquitoes produce the protective protein only in their nerve cells, the tissue that needs protection most. This second group does much better than the first in growth rate, survival, and pesticide resistance. Other kinds of mosquitoes also produce esterases in the intestine and the cuticle, where the insecticides enter the insect body. These may act like tiny organic robots attaching to the insecticide, making it harmless before it gets close enough to nerve cells to do any damage. This difference in the position in the body from which the esterase begins its defense is a continuing experiment in the evolution of increased pesticide resistance.